with, okay, perfect. So we're going to get started with our career panel. So welcome everyone. This is Black in Biophysics career panel. And we have a great panel today to talk a little bit about their journey in science. I'm Heather Pinkett from Northwestern University. I'm an associate professor here. And I'm going to introduce each of the panel members and then give everyone 3 to 4 minutes to kind of talk a little bit about their journey for this part of the discussion. So our speaker list is first, Dr. Barnes, who is an assistant professor at Stanford University starting in 2021. He's currently at Caltech. Dr. Alex Gorfe, who's an associate professor at the University of Texas Health McGovern Medical School. Dr. Garcia-Olivares, who is senior manager, preclinical pharmacology at Supernus Pharmaceuticals. Dr. Richardson, AAAS Science and Technology Policy Fellow, Office of Brain Director at the National Institutes of Neurological Disorders and Stroke, or NINDS. Dr. Okafor, Assistant Professor at Pennsylvania State University. Dr. Tolbert, Professor at Case Western University. Dr. Mario Penzo, Principal Investigator, National Institutes of Health. Dr. Curtis Hodge, entrepreneur at Nanoteam Technologies. And last but not least, Dr. Okeyo, Director, Portfolio Manager at NAN Ion Technologies. So welcome everyone. Thank you so much for being here. We're really excited about today's event because we just seem to have a wide array of different backgrounds and different types of jobs within the scientific realm. So first on my list is going to be Dr. Barnes. If you can just take 3 to 4 minutes and tell us a little bit about your career path.
Sure. It's a pleasure to be here today. I wanted to share a presentation, but screen share is enabled, so I cannot right now. Can we do it now? There we go. So I just want to quickly tell you about my journey, which, as Dr. Pinkett just mentioned, I will be starting as an assistant professor at Stanford in about a couple of weeks, which is very exciting. Let me just do it this way. So my journey started at UNC Chapel Hill. Back in 2004, I entered undergraduate with the aspiration of becoming an MD. I think like many people, and combining my academic prowess with football and social activities, that my grades soon took a hit. So I was like, okay, how do I overcome these poor grades? So I reached out to a chemistry professor and was able to join the lab of Dr. Gary Pilak. So this was my introduction to scientific research in Dr. Pilak's lab, where he studied protein-protein interactions using NMR. And so, like many people, I didn't know what research was. And so Dr. Pilak, Gary, he really instilled in me the idea of becoming a researcher and choosing this as a potential career path. So with this background I got in his lab in biochemistry and introduction to biophysics, I want to kind of explore more. So I went to the lab of Dr. Ron Montalero at the Center for Vaccine Research. This was very different, very new, because like I said, I never had done research before. And so my introduction was to biochemistry and biophysics, but obviously there's other fields. So I took a step out of my background in Gary's group and went to the CVR where I learned virology. And I think it was a really important step because what I learned was this wasn't for me. The fast-paced nature of biochemistry and biophysics was really what I enjoyed about science, not necessarily sitting and setting up cell cultures for 2 to 3 weeks before I could do one experiment. That wasn't my cup of tea. But again, I mean, that was a very important experience for me to understand as a emerging scientific researcher that I want to do something more biochemistry and biophysics related. So with that, I soon joined the graduate program at the University of Pittsburgh and joined a group of Dr. Guillermo Calero. He was a young PI. I was only his second graduate student. So, it was really exciting time to be part of a lab that was just getting started and to, really work, closely with Guillermo as he was able to teach me a lot about crystallography and really opened my eyes to other potential techniques, including collaborations with Tamir Golan to do micro-ED, working with groups at Stanford to really do engineering for and working with the X-ray free electron laser. So all these kind of techniques I was kind of exposed to as a graduate student in Dr. Calero's group. But working on eukaryotic transcription machinery, when you're writing grants and you always get to the health importance and relevance of these grants, we always were, just basically writing cancer, right? But in all honesty, we cannot, target the transcription machinery, or at least the basic level transcription machinery in cancer therapy. So I wanted to really take my skills and work on something that was more translatable. And so that led me to the lab of Dr. Pamela Bjorkman here at Caltech. Her lab focuses on HIV work and more recently on SARS-CoV-2. And so taking all these skills I learned throughout my career in biochemistry and biophysics, I was able to learn cryo-EM and really understand how to use structure-guided methods to design protein-based therapies as well as immunogens for vaccines. So with all that, all these experiences have really led to who I am now. And that's, soon to be assistant professor of biology at Stanford, working in the ChemH Institute, where I hope to use all these skills I've learned, including NMR, X-ray crystallography, cryo-EM, mixed with the underlying principle of biochemistry, which is necessary for a biophysics type of trajectory, to really understand viral hosts and our interactions. And importantly, throughout all this, I've had great mentors, and I think that's really key in any field you go into, whether academia or within industry. Having mentors that can really help you along the way is really important, and I've been lucky to have that. So that's who I am, and I look forward to answering your questions later on.
Excellent. Thank you, Chris. That was wonderful. And I really like the theme of try everything until something sticks. Awesome. So next up, we're going to have Dr. Okafor, who'll tell us a little bit about her journey.
Oh, hi, everyone. I don't have slides, but I'm really excited to be here. And thank you all for being here as well. I'll just, share briefly. So I went to a small undergraduate college, and I think that will be a common theme. A lot of us didn't know what research was until we somehow were fortunate to get exposed to it. And so I was fortunate to be able to go to an R1 institution to be accepted into a summer REU program. And during the program, I was able to do computational research, which is something I always say that was a pivotal moment for me, because before that point, I knew that I liked science, but I never had ever liked being in the lab, and I still don't like being in the lab. So when I did computational research, I was like, I can do research, but not be in a lab. This is transformational, right? And that was when I decided that I would go to grad school. And so I guess my one thing that I'll highlight there is that it's just exactly what Chris said, right? It's good to try many things and be open to options and just be open to opportunities, because you never know, you don't know what you like until you try it, and you just don't know what will stick, right? So that was, for me, that was the thing that helped drive me along this path. And so I ended up in grad school, did MD simulations, and grad school was kind of a windy, weavy road. I did a lot of things. I worked on RNA. My thesis was on RNA, but I always kept coming back to proteins and MD simulations, which is what I did in my postdoc. Postdoc is what you do after grad school. And as I started a postdoc, I was pretty sure that I wanted to pursue a teaching intensive position, because at that point in my life, the people that had been most influential in my life had been my undergraduate professors. So I was like, I'm gonna go and do this so that I will inspire other people as well, and give back. And so this is what I went into my postdoc with. This is the mindset that I went into the postdoc with. And I went into a teaching postdoc, which is where you spend some percentage of your time teaching and then the rest of it doing research. And during that time, what happened or what changed the trajectory for me at that point was that I ended up working on something that I really, really enjoyed. So up until then, I'd done research on different topics. I'd liked them, but I hadn't liked them enough to be like, oh, I'm going to do this, for long term. Then my postdoc, I was like, oh my goodness, this is fascinating. We don't understand it, and I'm going to figure it out, right? Or I have this unique perspective that I'm bringing that no one else is looking at it this way. So this is going to be my niche. And it was there that I decided, okay, maybe I should try and pursue a research intensive position. And so I went and talked to my advisor and I said, what do I have to do to make myself competitive for this? And we talked and we mapped out a plan, which included extending how long I thought I was going to be in the postdoc for 2 or 3 years. I think I ended up 4.5 years, extended that length of time and applied for funding, tried to get out papers, tried to do all the things that you do to become more competitive, which luckily we were successful. And now I'm here in my position. So my lab uses MD simulations, which I can talk about what that is. It's a computational method to understand how proteins are regulated. Very kind of high level explanation. But that is, I think I'll stop there and I will happy to answer any questions later.
Excellent. Thank you so much, Denise. I love this idea of like the aha moments. So, okay. So next we have Alex Gorfe, will tell us a little bit about his path.
Okay. Thank you, Heather and Lysandra and Whitney and the rest of you for putting this together. It was really time that we do this, and I appreciate the effort. I will start from slightly a different angle. I have to tell you where I came from. So I am from Ethiopia, from a small corner of Ethiopia, a small village. And I can talk on and on how it is and how it feels like to be first for everything, to go to university, to have a degree, to go abroad, to get a scholarship. But I leave that for a longer talk or even a book. But for now, I should tell you that I first studied industrial chemistry, but when I graduated, there was no industry to go to in Ethiopia. So I ended up in a hospital and I was a clinical chemist for 5.5 years, all the while writing letters to this and that university to get scholarship abroad. And I ended up in Norway, Tromsø. You can Google it. It's at the edge of the Arctic Circle. So that's from the tropics to the Arctic Circle almost. And I did masters there in physical chemistry. And what hooked me, what attracted me into that field, which was computational, which I mentioned earlier, but more on solving Poisson-Boltzmann type of equations in a physical chemistry setting. And what led me to that was a professor there, Arnas Malos, was trying to recruit us, incoming master students. And he was showing us these beautiful electrostatic surfaces on trypsin structure. And it was really, really beautiful. And I asked him, how did you get this? How do you do this? And he told me that, he solved some equations in computers. You can do that in computers. And I didn't know much about computers at the time. And I said, I wanted to do this. And I joined his lab and did my master's and then moved a little bit south into the center of Europe and did my PhD at the University of Zurich. But I always wanted to do both computational and experimental biophysics. And I started to do that, joined an experimental lab. I had somebody helping me out from the computational side, but doing it most of the time just on my own. And I figured that, to try to specialize on both was quite a mistake. It's very hard. So I ended up doing mostly computational work and did a postdoc with Amadi Okafor, quite a known person in the molecular dynamics and other computational drug discovery field. I stayed with him for 2 years as a postdoc and then came with a scholarship to the University of California, San Diego, and did additional postdoc with J. Andrew McAmon. Those in the field would know the name. And then I was working on a group of proteins called RAS proteins. I was just fascinated with them. And the reason for my fascination was really because I do not know enough about those proteins, how challenging they were to study, not just computationally, but also experimentally. These are surface-bound membrane proteins, highly dynamic, highly flexible. But we learned quite a bit simulating those proteins and then collaborating with experimentalists to interpret our results or test hypotheses. I'm still working on those proteins with some additional projects, and we have had some successes and some challenges. So I joined the University of Texas Medical School in Houston in 2009 as an assistant professor, promoted to associate professor with tenure in 2014, and recently promoted to full professor. So I'm still working on RAS proteins, and those I again ended up working on those kind of as an accident. I should skip the details, but life, can lead you in this kind of very, very tortuous paths. But ultimately, I would say to success. And I will be happy to share details on all or parts of this later on. Thank you.
Great, thank you so much. So next we have Jennie Garcia-Olivares, who will tell us a bit about.
Hello, everybody. Very excited to be here. And this morning I was remembering that Society for the Biophysical Society was my first scientific society where I participated when I was grad student. And my journey, I also have let me see, I'm gonna share this. Can you see it? Can everybody see it? Okay, then essentially this is my journey. I try to put together all the different transitions that I have done in my life. And there have been quite a few. I am the result of a lot of political movements. And I say that because my family is, and they are exiles from Chile, from the dictatorship in Chile in the 70s. Then they moved to Cuba. Then I consider myself Cuban, but my family is Chilean. But essentially, I graduated from college in Cuba, and from there, I start my own journey, let's say. And we went back to Chile, where I did my master's degrees in neuroscience. And from there, I joined a Ph.D. program at the University of Chile in a DAD program. This is a collaboration program between Latin American universities and German universities. Then I did my grad school in Hannover Medical School. And from there, I came to the US, first as a postdoc at the University of Pittsburgh, and later on as a research fellow on the intramural program at the NIMH. What I have in common between all these different projects and places is that I always work with membrane proteins in different capacities. I start my master in AMPA kinase receptors, then I moved to calcium channels and then to chloride channels. And during my postdoc, I went a little bit more deeper into pharmacology and tried to understand the effect of psychostimulant. And for that, I work with monoamine transporters and glutamate transporters, GPCRs, and generally G proteins. Then how do I end up being in industry? That was completely out of my sight. I never saw myself as moving into industry. But after I was finishing my projects at the NIMH, I kind of got, I don't want to say bored, but somehow in that loop that you have your project, you invest all the time, you have your paper, and then after the paper is published, you get into that empty space that you have to start all over again in the cycle. And somehow I didn't want to spend all my life going kind of in the same sub questions or the previous questions, that I found that sometimes academic life is kind of infinite loops of knowledge that you want to fill. And actually, I am in industry because random luck. I was, as I said, in that space, my space, that I wasn't sure about what to do. I wasn't sure if I want to do academics. I have a viable project, a good place to be, but I was not completely fulfilled. Then I found this mid-sized company that was local, then that helps a lot because I didn't have to deal with the idea to move. And that was that position open, that was preclinical pharmacology. And I applied, and they were working on an ADHD project, working with my transporter, with the dopamine transporter, dopamine transporter. Then I got the position. I have to say I have been there for 2 years and has been a wonderful experience. I'm very happy of the transition, all the exposure that I have got in these 2 years, all the different aspects of the job itself. Just to clarify, we don't have wet labs. I mean, essentially we contract out all the preclinical studies to different CROs. Then that allows us to do many different things. And I think all this background in ion channels, in glutamatergic synapses, I mean, all my experience has been actually very important in this transition, because our projects go from epilepsy to migraine to ADHD. We just got a 1-hour drug was approved for the FDA for ADHD. And to be part of that regulatory process, to be part of that development of a drug process, I mean, has been really amazing for me. Yeah, I don't want to say more. We can expand a little bit more later. That's it.
Excellent. Thank you so much. I like this idea of unexpected opportunities. Okay. So next up, we are going to have Dr. Penzo tell us a little bit about the journey.
All right, can you guys all hear me?
Yes.
Great, so it's really a great pleasure to be here. Thank you so much for the invitation. So I'm a tenure track investigator at the National Institute of Mental Health. I'm not really a biophysicist, I'm a neuroscientist, and my laboratory studies essentially neuromechanisms that help animals cope with challenges that they encounter in their environment. And we essentially do this by looking at the activity of neuronal circuits, but also at the cellular and molecular mechanisms that shape up the activity and the functions of the circuit. So some aspects of our research actually deals with things like the biophysical properties of the neurons, and actually part of my path in science has been inspired by great biophysicists, and one of my professors actually trained as a biophysicist from grad school. And so anyways, that's sort of my link to the biophysical community. So I'd like to tell you a little bit about my background. I'm actually also from not from the US. I was born and raised in the Dominican Republic in the Caribbean, and I moved from my country and to Puerto Rico when I was a teenager, so essentially at the age of 12. So I did my high school and I did college at the University of Puerto Rico. And it was there when I was at the University of Puerto Rico that I had my first encounter with science. You know, like many of you, I had this idea when I was in high school that, you know, I like science, so I wanted to go get a medical degree. And usually, you know, when I was in high school during the summer times, also early on in college, I'll spend my summer trying to get money doing some jobs, like, you know, like working at a restaurant or movie theater. I did a lot of different things, supermarket. And when I actually was like in my junior year, and I had this opportunity that was offered to do research for the summer and actually get paid while like having fun doing experiments. I was like, really, you can actually do that? I mean, I'm here like working so hard every summer doing something that I don't enjoy so much just to get some money. So if I could have fun doing science and getting paid for it, that's a dream. So that was like my you know, real awakening, you know, because when I started to get into science, it was clear to me that there's so many things like like somebody else said recently, Denise, I think, how you know, so many things that we actually don't have an answer for, and the brain is very peculiar in that regard because we very limited knowledge about the brain, and as a result, we struggle to find solutions for things that we care a lot about, like psychiatric disease, particularly here at the NIA. So I did spend some time while at the University of Puerto Rico working for my last 2 years in college with Pedro Frisbee, who actually is a biochemist, but he studied neuroprotective mechanisms in the hippocampus, and this sort of like got me. This was the first experience that got me interested in brain research, and I'd like to use this opportunity to discussing my time in doing research in college to highlight the importance of mentors in our careers, like Christopher mentioned earlier, because I think it's really, I think what I find is the common denominator, in having a successful path in science, is having mentors that not only serve to like inspire you and sort of like motivate you to pursue a career in science, but also provide that kind of like support, constant support. And that was something that I was very fortunate to get on quite early during my time as in science. I was blessed in that regard, and that sort of like was a recurring theme. When I went to grad school, I moved from Puerto Rico to New York City. And I got my Ph.D. from Albert Einstein College of Medicine in the neuroscience department there. And shortly after, I completed a postdoc also there in New York at Cold Spring Harbor Laboratory. And I left New York City and started my position at NIH in 2015. And as I said, I'm in the tenure track here at NIMH. So I'll stop here and I'll continue later in the discussion, the Q&A.
Great, thank you so much, Dr. Penzo. Next, we have Curtis Hodge.
Hi, thank you. I also have a little presentation. Okay. Can everybody see that? I can't see your faces.
Yes, we can.
Okay, good. Great. Thanks a lot for the invitation to come speak. I think I might be the only sort of startup scientist on the panel. I'm not sure, but yeah, so I will be happy to answer any questions during the panel discussion about starting a startup, things like that. But for now, I will give just a brief overview of my sort of career trajectory. So I like to put the pictures of how I change my appearance because it kind of tells the story, I think, a little bit of the light in your eyes going from one stage to the next. But yeah, so I started out, so actually I'm Canadian. So I started out in Canada. I did my grad school at a place called the University of Alberta. And it was on using X-ray crystallography, small angle X-ray scattering, looking at DNA repair proteins, ubiquitination, specifically enzymes. And also I got some experience doing some cell biology there as well. So that sort of, fueled my interest in using proteins and understanding sort of fundamentally what drives a cell to do what it does. So, understanding DNA and proteins, all that stuff. And then I, to take a step back, sort of one of my primary motivators has always been to try and have an impact on, cancer therapies and, trying to improve those. And so I could see during grad school that immunology was going to play a really big role, that it was sort of an largely untapped area for developing therapies and treatments. So I wanted to learn more about cancer immunology. I didn't, have much background in that. so I went to UC Berkeley for a postdoc, and that was in a cancer immunology lab. It was largely innate immune system focused, looking at CGAS sting pathway, things like that. But it was a real switch in sort of techniques. I went really from sort of a biophysical, background and into a very mouse model driven field. And so that was a significant change. I found, just like one of the other panelists, I found that I didn't really like doing mouse model work. So I wanted to get back to sort of the biophysical biochemistry area, but with an eye towards, cancer immunology. So I went and did a postdoc at Lawrence Berkeley National Lab. So anybody who's collected data at the Advanced Light Source, for example, I'm basically, my office is right in the outside of the synchrotron ring. And the idea there was I joined a group that they ran both an X-ray crystallography beamline, but also a heavy focus on small angle X-ray scattering. And the reason I did that was because I realized that it was a really great readout for understanding changes that you're making to a protein, right? X-ray crystallography, you're dependent on getting protein crystals, things like that. That can be slow. You can never end up getting them. Cryo-EM is starting to tackle some of that, which is great. But the thing with protein engineering, especially when you're trying to design macromolecular complexes, is you really need sort of a readout of how are you changing this structure. So small angle X-ray scattering is really quite high throughput. Well, there is high throughput SACs, but also size exclusion chromatography coupled SACs. So it allows you to look at proteins, complexes, very quickly, get a read on what you've done, how the complex has changed, and you can do a lot of cool modeling and get really good insights into sort of how dynamic your system is. So I was able to sort of harness that with my background, also, with the goal towards cancer immunology, and worked on developing these self-assembling protein nanoparticles into something that could be used. And so I basically was able to engineer that to bind and sort of aggregate or orient antibodies in a sort of a particular way that sort of clusters receptors. So the idea is to drive the activation of cells through clustering receptors. So I was able to do that. And Lawrence Berkeley National Lab supported a patent filing on our behalf. And so I decided, hey, you know what? The only way that science, really sort of new things, get really pressure tested and translated is actually through startup companies. And then those often get acquired by larger pharmaceutical companies or licensed, things like that. And I figured if I didn't do it, no one was going to do it. So I decided I was going to learn how to be an entrepreneur and start a startup. So I did that, and able to get traction, a lot of hustling. So anybody in the audience that likes hustling, that's largely what the entrepreneurship is. Talking to investors, doing pitches, things like that. And so that's sort of where I'm at. I was able to get us significant investment, and we've just opened a lab in Berkeley. And I'm actually right in the middle of transitioning full-time to working for my company. We're also hiring. So anybody who's got a background in T-cell biology, just let me know. But that's sort of where I am and what I've done. I'm happy to answer any questions and go into any detail about what the entrepreneurship game is like. And with that, I will stop.
Excellent. Thank you, Curtis. Okay, so next up, we have Dr. Blanton Tolbert, who will tell us a little bit about his journey.
Okay, so first I'd just like to thank the organizers for this panel discussion. I'm really excited to be here. And also I'd like to thank the organizers of this affinity group for what I think is really creating a safe space for us to have these conversations. So I'm from a very small town in South Carolina. I did my undergrad at the University of South Carolina in the Department of Chemistry. Like I think several people here, I started out in undergrad thinking that, I wanted to go to medical school. I was a bio major, and after my freshman year, I realized that I have to take way more chemistry classes to get this biology degree. So I decided to switch my major to chemistry. And so I switched my major to chemistry after my freshman year. And, that was a really good decision because it got me involved in undergraduate research. I started out really just working in a stock room, general chemistry stock room, but then I was able to find a home in an analytical chemistry lab. And that lab was, really using like Raman and IR spectroscopy to analyze bodily fluids. And so I did a lot of buffer preps, and I became really good at making buffers using the Henderson-Hasselbalch equation, enough so to realize that I did not really enjoy analytical chemistry. So I graduated with a chemistry degree, 99, started a family at the same time. So I had my oldest son, who's 21 now. And I wasn't quite ready for the PhD. And, I was kind of in a transition period. So I went to Hampton University for a couple of years, and I kind of wanted that HBCU experience because I didn't have it. And I spent 2 years at Hampton where I really focused in, it was a master's program, where I really focused in on deep diving in chemistry. And so it really gave me sort of this opportunity to just sort of think about what I wanted to do in chemistry. And I got interested in biophysics there. I did a summer program while I was at the University of Rochester, and I started working on this family of model peptides or cytochromes known as microperioxidases, and doing ligand binding studies and some computational studies as well. I ended up at University of Rochester in a biophysics structural biology program. I was in that program. The lab that I was in, the guy, his name is Ravi Basabappa, a phenomenal guy, great mentor. His focus was X-ray crystallography and studying enzymes and ubiquitin proteasome pathway. About midway through my PhD, Ravi left the university, so I had to find another home, and that was a tough period, and I can talk about that a little later. But I was able to find another home, and that home was in Doug Turner's lab, who is very famous for developing rules for RNA structure prediction. So in that lab, I started to use NMR spectroscopy to work on RNA. And I knew at that point, right, that I wanted to, in graduate school, I had this realization, this kind of epiphany that I wanted to run a research lab, in part because of, some of my experiences at the time. But I had this epiphany that if I do this, I want to actually combine these two areas, this stuff that I started working on in the protein world and doing protein biophysics and RNA. So I was luckily fortunate enough to be sitting at a seminar when Mike Summers came through to give a talk at University of Rochester, and at the end of his talk, he showed pictures of the Meyerhoff program, and I was like, oh man, what is this about? I didn't know anything about it, and so long story short, I wrote Mike an email and said, hey, I'm going to be graduating. Can I come do a postdoc in your lab? He accepted me as a postdoc, so I went to learn more advanced NMR techniques to solve RNA structures of viral RNAs. And that's kind of where I got my interest in RNA and related retroviruses. And so that's what we do today. I started my independent career as assistant professor 2009 at Miami University of Ohio. Smaller school in Ohio, but it offered a PhD program in biochemistry and several other PhD programs, and had amazing magnetic resonance facility, so I could really do some high-end NMR. I was moved to Case Western pre-tenure 2012, promoted to associate 2015 in full, 2019. So, I run, and, I'm proud to say, an NIH-funded research lab. We study really the biophysics of protein RNA complexes with the goal of trying to understand how those complexes regulate gene expression. I'm very much interested in integrating biophysical methods and sort of really rigorously, in my opinion, characterize these sort of systems. And so that's what we do. I have a really diverse group of trainees in the lab, from high school students all the way through to postdocs. And so it's been a really great ride, and I've had a good time with this career choice. Thanks.
Excellent. Thank you so much, Blanton. Okay, so next up we have Dr. Okeyo. He'll tell us a little bit about his path.
Hello, can you hear me?
Yes.
Excellent. Thank you very much. I really appreciate being here, and I thank the organizers for this opportunity to be speaking to you all today. And looking at this forum, I wish that back in the day when I was in grad school or a postdoc, that I had exposure to such a forum. So I'm really grateful for it, and I'm hoping that the audience will have something to take home as regards careers. Currently, I'm the Director of Portfolio Management at Nanion Technologies. This is a biotechnology company that is specialized in building automated patch clamp electrophysiology systems. And these would be spanning from low to high throughput systems that are used in drug discovery. We also build other technologies such as instrumentation for in vitro contraction force measurements in cardiac cells, for elucidation of responses of these stem cell derived cardiomyocytes, and other platforms used in safety screening, generally the safety screening space. We work in partnership with academic institutions, pharmaceutical companies, both large and small, big pharma as you might call it, and contract research organizations. And one might wonder, so what does this entail, me being Director of Portfolio Management? To keep it brief, what I do on a day-to-day is I'm involved with the development of corporate strategy in business development, project management, and overseeing teams that are involved with different aspects of our business. And so, this is what you'd categorize as a non-traditional role for a scientist, whatever that means. But, to be clear, everything that I've accomplished as a scientist, every publication, every conference presentation that I've ever made, every class I ever taught, all my research projects have been instrumental in equipping me to be successful and to really make me what I am today. So, looking back, my career journey has taken me through very many different roles, research and development, sales and marketing, management of customer relations. This is spanning from bench science to even more business-centric roles, and things that I never really knew were possible before I got introduced to this world. So how did I get here? My science journey started in Kenya, which is where I'm originally from, at the University of Nairobi, where I studied chemistry as an undergraduate. And with my freshly minted degree, I went into the workforce, joined Bristol Myers Squibb there as a pharmaceutical sales representative. And what I was specialized in was what they called a anti-infectives expert. That's what I was. And I was also involved in the cardiovascular therapeutic space. And the job involved interfacing with physicians, updating them about drugs and treatment options, and day-to-day care of patients and so on. And this was really the beginnings of me getting into research, because what we used as tools for the job were clinical study reports and efficacy studies and so on. And these were the tools we used to engage with physicians. And so from there, I got this inspiration for graduate school, because I always thought to myself, wow, I mean, this is amazing, the kind of work documented here. And I just had this dream of, being an author of one of these. And that's how I started applying to graduate school and got admission at the University of Florida, and started my graduate education there. And at that point, I got introduced to the world of ion channels and biophysics in general. And that's really why I'm doing what I'm doing today. So during grad school, I worked on protein purifications of the large conductance calcium activated potassium channels, mechanosensitive channels of large conductance. But all these were reconstituted proteins in biomimetic membrane systems, or what you'd call artificial membrane environments, bilayer experiments. That's the kind of thing I was doing. And going through all this, and because of my pharmaceutical background, I thought of being focused more on what I thought to be biomedically relevant. And after grad school, I decided, well, I'll pursue a postdoc that would bring me closer to what would be, say, more relevant to treatment options, and something that I felt was more practical or closer to helping patients. So I started my postdoctoral studies at the Cystic Fibrosis Center at the University of Alabama, Birmingham, and worked on the cystic fibrosis transmembrane regulator protein, which when defective, this is what causes cystic fibrosis. And this now got me more and more into mammalian systems and more of ion channel physiology and biophysical characterization, and studies of these. And I think at that point I felt, well, I'm getting closer to what my dream always was. So after my postdoc, I joined Nanion Technologies, where I've been for the last 10 years. I started off as an application scientist, involved with helping users of electrophysiology systems, assay development, problem solving, and ending up with interfacing with customers, which was related to a large degree to what I was doing when I just came off of my undergraduate studies. So this is the path I've been on, and it's a little different, I would imagine. And I think in totality, everything just fell in place, and I think every little aspect, much as it felt not related at that point, really has amounted to what I am today. And I'd be glad maybe to answer any questions at the Q&A session. Thanks.
Great, thank you, George. And last but not least, we have Dr. Ryan Richardson. Tell us a little bit about your path.
Thanks for having me on this panel. It's really an honor to be here, especially among so many really accomplished peers. It's really a great opportunity to talk about some of my background. So let me share my screen and start my timer so I don't go terribly over. So in thinking about my path, what I really wanted to start off with is thinking kind of broadly how I see myself as a scientist and how I see the role of a scientist in general, because this really is flavor a lot of my decisions along my career path. So I think really broadly about science and engineering as mindsets, and I'll get into a little bit more about what I mean by this. So thinking about my academic and research background, I'll go into this a bit more in a few slides, but I've worked in a variety of different projects and organism systems, worked in virology, worked in neuroscience, cancer biology, oncology, I've worked in a variety of different cell systems, insect cells, worked in mice, and worked with human cells using a variety of different substrates. DNA and protein are my particular favorites, but also working with chemistry, chemistry tools, and also computational tools as well. And why I mention all these things is I'm kind of a jack of all trades, master of very few in a lot of ways, but what really kind of united these different pursuits over the years has been really an emphasis for me on problem solving. And that's really how I see myself as a scientist. I see myself first and foremost as a problem solver. And I love thinking about new and interesting challenges in biology and really developing new tools and new ways of addressing them. And my particular form of problem solving has come in the form of synthetic biology. So very briefly, synthetic biology really is the design and construction of new biological parts, devices, and systems, and then kind of redesigning and engineering existing natural biological systems for useful purposes. So thinking kind of more broadly about biology as programmable units, thinking about it kind of as a circuit board with the inputs and outputs being biological parts. So thinking about how we can engineer DNA and RNA and proteins as well to come up with new biological functions. I won't talk about some of my more specific work, but just to say that I've worked on a variety of different synthetic biology projects, gene editing, both in vivo and in vitro, and I'm happy to talk about some of those. So again, thinking more broadly about science and engineering as this problem-solving tool set, really. Thinking beyond the individual tools and techniques that we learn along the way, and thinking more broadly about some of those intangible skills, right? The analytical, critical thinking, and experimentation skills. Because that has led me to realize that this has been an incredibly unexpected journey for me, and I would not have been able to predict it from the onset. And part of that is the understanding, again, that problems exist everywhere. So I'll speak a little bit more about some of my research and really the focus on understanding human biology. But that has led me to think kind of more broadly about how I can have an impact using my skill set as a biologist, as a scientist. And that's led me to forays in business, thinking about business consulting and some challenges there, but also more recently into the policy realm. Right, so the obligatory Gantt chart. So I started off my research career at Northeastern University. I did got a bachelor's of science in biochemistry. I was actively involved in undergraduate research in a couple of different labs. Northeastern also afforded me the opportunity to do internships in the local Boston area in biotech and pharma companies. So it was a really great opportunity to both understand what I did like about science and what I didn't like about science. After graduating, I got a position as a research technician at Mass General Hospital in Boston. And this was a really transformative experience for me. Up until that point, I didn't really have a great understanding of what it meant to be an academic, what it meant to have a career in academic science. And getting to work with really talented investigators, grad students, postdocs, and also interacting with the principal investigator of that lab really established for me that that was something that I was really incredibly interested in. So that led me to my Ph.D. in biochemistry at Harvard. My PhD was a bit of a meandering path. I think I did about 6 or 7 rotations, testing out different labs before ultimately deciding on the synthetic biology lab that I chose. And again, I won't talk about many of the details there, but ultimately through those 5.5 years, I started to get the understanding, at least for myself, that I didn't see myself kind of following that traditional academic trajectory. And I didn't really envision myself as a principal investigator running my own lab for a variety of different reasons. So I started thinking about different options out there. And one of the options that I got particularly interested in was thinking about business consulting. So again, thinking about problem solving just with a different set of problems to engage with. And I thought there are a lot of really interesting challenges in business development, particularly thinking about, again, how we can have a broader impact using the research tools that we've developed over the years. I ended up doing a postdoc, kind of in name only, at the University of Maryland, Baltimore, working with a good friend of mine to help start up his lab. And after 3 years there, I kind of came to another decision point again and had to figure out what to do with my life. And have kind of serendipitously fell into this science policy fellowship. So this is a fellowship through the American Association for the Advancement of Science. And the focus is really on placing PhD scientists and MDs in the federal government in a variety of different agencies to be able to use their background and skills to further the goals of policy. I am currently placed at NIH, National Institute of Health, with the BRAIN Initiative. So this is the brain research through advancing innovative neurotechnologies initiative. At the NIH, at the BRAIN Initiative, I perform a variety of different roles on the programmatic side with implementing different funding announcements, helping out with making those funding decisions and reviewing the programmatic goals. And also program evaluation as well. In addition to that, thinking about kind of the more strategic level of communications, outreach, stakeholder engagement, and something that I am very passionately involved with are also diversity, equity, and inclusion efforts. So again, thinking about the tool set that I have and how I can have a broader impact with those tools. All right, so again, it's been a very winding journey, and I think I often had hoped that it would be something linear like this, but it's more turned out to be a very winding and circuitous pathway, which is okay. The last thing I want to end with quickly is that it's really important to have fun along the way. I think it was Dr. Barnes that mentioned in undergraduate playing football. And for me, competitive sports have really been a tremendous outlet over the years. And throughout grad school and before grad school, I've been able to compete at a very high level at a semi-professional, professional, and ultimately winning a world championship in ultimate Frisbee. So this has been really tremendous. And it's just to say that you can be a scientist and be multifaceted as well as that. STEM education and outreach have also been really strong passions of mine. And I've been involved in a variety of different efforts, both locally in the Boston and Baltimore area, but also internationally, working with some programs in China and also Mexico as well. And with that, I will end things up. Happy to answer any questions along the way.
Excellent. Thank you so much. So thank you to all of you. Despite the diversity in our paths, I do see some common threads. So I'm really excited for us to get into breakout rooms and have a discussion about some of these commonalities that are in our path. Before we get started with our breakout rooms, I really want to give our second co-host the opportunity to introduce herself. So hi, Theanne.
Hi, everyone. I'm so sorry that I had to join late. I actually had a different event during the introductory portion of this that ended a little bit early. So because we're running late, I will just very briefly say that my name is Theanne Griffith. I'm an assistant professor, just opened my lab at the University of California, Davis in October. I got my PhD in both neuroscience and Spanish from Smith College in 2008. And there I was fortunate enough to do research in all 4 years. And that's kind of where I got my introductory taste into ion channels. I studied human GABA A receptors and was pretty lucky to learn 2-voltage electrode clamp as an undergrad. And so I did a lot of pharmacology experiments with novel anesthetic compounds there. I then took my knowledge and love for Spanish south of the border, all the way south to Chile, where I worked for 2 years in a lab at the Catholic University there doing Alzheimer's disease research, kind of stepped away from ion channels and biophysics for a bit before returning to Northwestern University, where I jumped right on back onto the biophysics ion channel train and studied structure-function relationships between glutamate receptors and their auxiliary subunits with Jeff Swanson at Northwestern. following that, I wanted to add, so full disclosure, I absolutely love biophysics, but my research really does kind of walk the line of biophysics and physiology. I love taking what I know about how ion channels work and applying that to physiological context. So I joined Ellen Lumpkin's lab at Columbia University, where I got really into somatosensation. And now currently what my lab investigates are the ion channels and receptors that are involved in the transmission of thermal signals as well as thermal pain signals. So we use a combination of approaches, electrophysiology, behavior, even some molecular profiling. But I absolutely love ion channels. I love understanding how they work. I love understanding how they contribute to neuronal function. In addition to my research, I'm also a children's book author. I write the science adventure series, The Magnificent Makers, which again is what I was literally just presenting right before hopping on to this call. So I'll just leave it at that. I know that was very brief and not detailed, but I can answer any other questions if anyone has them in the breakout sessions.
Excellent, thank you. Okay, so I'm gonna rely on Whitney and Lysandra to guide us into our breakout rooms.
I believe we have to sign on to the new links, correct? There should be links for A and B that everyone received.
Excellent, thank you for putting them in the chat. Okay, so right now we are going to go to our individual groups. I can tell you the breakdown of the A group and the B group. Oh, and it's listed in the chat too. So for the panel itself and everyone attending this event, we will see you in our breakout rooms. Thank you, everyone.